Thanks, Ed. Good afternoon. Welcome back. As mentioned, I'm in the Center for Drugs. So what I'll be speaking to you about is really the drug development perspective rather than the device development and clearance or approval process. So I'd like to first also thank Tal and the rest of the organizers for the invitation and organizing what I think has been so far a very excellent meeting. So one thing I'd like to say is first, I'm clearly not able to cover all of the different activities that the agency has ongoing in the personalized medicine space in a 15-minute time span. But you may have seen a report that we recently issued which really catalogs the myriad activities of our organization and what we've done in this space on the science and technology front, the research front, as well as the regulatory front. And I think to me this captures really the bulk of what we do. Now one thing I think that really is important to me and is a message worth reiterating is that personalized medicine, insofar as it's the practice of medicine between a physician and a patient, is always personal. And what we're trying to really do is help facilitate the transition of new technologies to patient care through clinical trials and targeted therapeutics. So yesterday we heard a number of different retrospective looks at personalized medicine over the past 10 years. And if I was going to capture what the agency was doing 10 years ago, first of all, I wasn't there, but I think Drs. Lesko and Woodcock have been pioneers in this space and really sort of set the charge for regulatory approaches to personalized medicine, specifically in the form of pharmacogenomics and targeted therapy development. So many years ago, they basically opined that there was a major gap between what was being done in terms of research and its actual application in the clinic. And I would posit that that gap still remains. I think there's a number of drugs that we have available which could have personalized approaches that we clearly aren't using in the clinic. And this is definitely not a new concept. Our office, my office, is the Office of Clinical Pharmacology. Our core business day to day is personalizing therapeutics. And we do this in many different ways. We do it through drug-drug interactions and dosing recommendations, which patients should and should not get therapeutics. And the way we look at this is genetics is really just another variable in that mix that explains variability in drug pharmacokinetics or drug response. And speaking to this point, we have upwards of 60 or 70 molecules that have genetic information included in labeling. And the recommendations for that range anywhere from no actions, just informational, all the way to do a genetic test if you reach a certain dose threshold, and then certainly prospective dose adjustments or patient selection. So there's a very broad range of how genetics can be applied. And we've always taken the strategy that there is a base assumption that genetic information at some future point will be available to the prescriber at the point of care. But I do think over the past couple of years, we have seen that this gap is narrowing a little bit. We now have upwards of about 30 oncology compounds that are indicated for a very specific molecularly defined subset of patients, and this could be for predictive reasons, diagnostic reasons, or prognostic reasons. But I think what's interesting with the approvals that we've had in the past 2 years is that we've actually come to a point where we're now having companion diagnostics co-developed and co-approved and available at the time that the therapeutic product is approved. So I think that this is a major change that's been a long time coming. And we're now embarking on a whole new set of challenges. In the oncology space, high-resolution molecular profiling is becoming obviously much more common, as we've heard over the past couple of days. And in earlier phase clinical trials, we're beginning to see grouping of patients according to molecular characteristics rather than histological characteristics. And this, for all intents and purposes, I think is a very good strategy. You have targeted therapeutics that have a specific mechanism of action. It only makes sense to study those patients who are most likely to benefit from treatment, and often that's based on experimental evidence. We're also seeing larger scale master protocols, which I think is really starting to streamline the process of diagnostic testing and matching to a clinical trial for patients. So you can now have blanket testing procedures, put patients into a clinical trial that's specifically suited to their tumor. And this is very valuable in the early phase space when you're trying to explore and establish proof of concept, but we're also now seeing this in later stage phases to support approval for drugs and diagnostics. So it's going to have its own set of challenges, but it's something that we're in the process of dealing with head on. Now, as I mentioned, this is all a very long learning process. We've had a decade now of experience, and our experience continues to grow. And some of the things that we have learned over the years have been very instructive and distilled down into guidance and policy. So I think we've all seen that the evidentiary standards for retrofit biomarkers are very challenging and nuanced, and there hasn't been really any clear agreement on how one goes about retrofitting a biomarker to a drug that's been approved and in use. And warfarin is an obvious poster child for that, not the best example for personalized medicine. It's a drug that's been used for decades. Additionally, we've also learned that if a drug is effective in the overall population, you may not need a companion diagnostic. We do take risk-benefit very seriously, and it's a very important consideration to determine whether or not a drug should have a narrow indication versus a broad indication. But nonetheless, testing can still be used, even if not qualified as essential for safe and effective use, as our companion diagnostic guidance stipulates. Additionally, an issue that seems to be a recurring theme, but unfortunately still handled on a case-by-case basis, is the issue of whether or not you need to put marker-negative patients into clinical trials. And this is something that we've dealt with in the enrichment guidance and are continuing to deal with it as we go through the revisions to that guidance. But we have seen that labeling ultimately does depend on the studied population, if it's enriched or not. One thing that's becoming more of an issue as we have these higher resolution sequence-based diagnostics is actually the many shades of marker positive that exist. So for example, BRAF mutations, V600E is not the only one. Dealing with K and D and all of the other rare mutations in a clinical trial and drug approval context is becoming more and more of an issue as these technologies are making their way to clinical practice. Now, I've said we've handled some of these things on a case-by-case basis. Yes, that's true, but there are some emerging themes that do come out of these development programs. And if you look at the appropriateness of biomarker-based indications, there's a couple of factors that seem to be driving whether or not that's an appropriate pathway. One is whether or not the drug is intended for a serious and life-threatening condition with no available therapy. Clearly, FDA has had much greater flexibility in those contexts. And in fact, we even would endorse personalized development strategies in an effort to get those drugs to market faster and sooner to treat patients. Additionally, therapeutic risks and benefits are a major consideration. If a drug is potentially effective in the overall population, even if the biomarker helps refine that use, it still could potentially be approved provided it has a safety profile that's acceptable. On the flip side, if there's a drug that has toxicity concerns or long-term chronic poor outcomes, that might be a drug that would be more amenable to a targeted approach. Perhaps most important is the actual merits and limitations of the biomarker itself. Clearly, if you have a biomarker that has established medical relevance, it's much easier to pursue a targeted development strategy for those types of biomarkers. They have established utility. We understand what the medical decision-making is around it. And additionally, we also have to consider the various intrinsic properties. We have a lot of continuous biomarkers that aren't necessarily a discrete yes or no result. And some of those may be highly variable, and those are things that have to be worked out in the drug development process. But to the extent that we know that, it makes it easier to pursue this type of development strategy. So we've tried to take this experience and distill it into digestible guidance for the industry. And I have just a couple of those listed here. One is the Clinical Pharmacogenomics Guidance, which deals with early phase studies and the collection of DNA. We have the Enrichment Guidance, which is currently in draft form, which talks at great length about the targeting of specific subgroups of patients, whether it be for predictive reasons, prognostic reasons, or just to take the noise out of a clinical trial. Many of you are very familiar with the Companion Diagnostics Guidance, so it goes without saying. And we're in the process of tweaking the co-development guidance, and hopefully that will be released in the near future. So you can go to our website for all of the guidances and read at your convenience. A couple of other notable developments in 2013. We had a final rule that amended the orphan drug regulations, which basically defined an orphan subset of a common disease, which is something that's very important in the context of personalized medicine. If you take a common condition like breast cancer and narrow it down to a molecularly defined subset, you have different incentives that are available to you. So this is basically defined as use of a drug that would be inappropriate owing to some toxicity, the mechanism of action, or previous clinical experience, and it would be inappropriate in a certain subset of patients, but not the one that's being targeted. Additionally, we've developed the Breakthrough Designation Program, and this is a program that's designed to give drug developers very continuous advice in the drug development process with commitment from senior leadership. And as we're seeing in this program, there is quite a bit of targeted therapy development, as you would expect. Treatments have very large effects. They're targeted to very clearly defined molecular subgroups of patients, and about 3/4 of the designations that were granted as of last summer had some biomarker targeted component to them. And as has been mentioned a couple of times, next generation sequencing platform was cleared, and this has obvious implications for delivery in the clinic. And one reason I think that this is important is really with the issue of optimizing therapeutics from a safety standpoint. Very often, adverse events that are very severe in nature tend to be rare. However, we have a number of those where we've identified genetic markers that predict risk, but there's been no translation to the clinic, at least in the United States, and that presents a challenge because it's very difficult in the current drug test paradigm for a physician to order 1,000 tests to prevent one event. You just get fatigued and it doesn't happen. So this switch to a preemptive model through these next-gen sequencing tools hopefully will facilitate broader uptake of these, use of these tests for eliminating patients who are likely to have toxicities. And there's a couple of examples that we've had this year. Valproic acid is known to cause fatal liver failure in children who are very young, under 2 years of age. And some recent studies have shown that this traces back to mitochondrial dysfunction as a result of POLG mutations. So we have a recent contraindication to use that drug in these patients. Additionally, codeine was contraindicated in children who are undergoing tonsillectomy because there was reported deaths that were potentially attributable to CYP2D6 genotype. And it was very difficult to recommend testing in that situation because it wouldn't really adequately take out all the patients who could be at risk. So as a consequence, the drug was contraindicated in the broad clinical population. Now that speaks to the fact, I think, that if you have a drug with known liabilities, access to information at the point of care rather than a physician ordering a test could have much different implications for the risk-benefit profile of a drug in the post-marketing setting. And this transition will also be important. We have a number of post-marketing commitments and requirements where pharmaceutical industry is conducting studies to investigate genetic predictors of differences in drug pharmacokinetics or adverse events. And this will really set the stage for application of the findings from these efforts. So to wrap up, I'll just make a couple of quick points here on organizational activities. So at the agency, there's obviously multiple nodes that are involved in personalized medicine development. You've got the Center for Devices, Center for Drugs, as well as the Center for Biologics. And we meet on a monthly basis and communicate pretty much seamlessly throughout the course of drug consultation on drug development programs. So there is quite a bit of communication within the therapeutic product centers. More broadly, we have a number of groups that are agency-wide dealing with issues related to emerging technologies such as next-gen sequencing, imaging, and other modalities that are being used in patient care and other environments. It's also worth mentioning that with the recent reauthorization of the Prescription Drug User Fee Act, there's support for biomarker-related activities at the agency. So we are actively in the process of training our staff on the regulatory issues around personalized medicine development. Not everyone at the agency is an expert in it. We're also in the process of putting together a public meeting, which will likely be held later this year, and hiring folks who have biomarker development expertise. So to conclude, I think that we're at a place where we have technologies that are there. We're close, very high throughput. They're being deployed in the clinic. We've got drug development programs that are becoming more rationally designed, at least in the setting of oncology. These tests are becoming available at the clinical interface, which is important in allowing it to transition, whereas in the past we may not have had cleared tests and relied on laboratory developed tests. And we're also transitioning, at least in some centers, to this more preemptive model of testing, which I think is doing a lot for making decisions at the clinical interface and allowing prescribers to act, rather than having to order a test. There are obviously many challenges, least of which is how to get paid for this to bring back that return on investment. And also in a global drug and device development context, the regulatory certainty and burden are clearly issues that are presenting a challenge and will continue to evolve and have greater clarity over time as we continue to acquire experience. So with that, I'll conclude.
Thank you so much, Mike.